Earthworms burrow through the soil, taking up a lot of dead plant material. When genetically modified Bt maize is grown, these useful soil inhabitants could also ingest the Bt protein produced in the plants. Eva Schultheis from the University of Aachen has investigated whether this has any negative effect on the animals. Earthworms are essential to conserve the structure of the soil and its function. This is why when we talk about biodiversity and the effects caused by Bt maize on agriculture, the earthworm is an important object of study. The Bt maize used in this project contained 3 different Bt proteins. The maize was cultivated in a field trial and compared with a number of conventional varieties. During the 3-year experiment, Eva Schultheis and her team regularly recorded the earthworm population in spring before the maize was sown and in autumn after it had been harvested. This was achieved by digging up a predefined amount of soil in each of the 40 trial field parcels and searching it for earthworms. The animals were preserved in alcohol and later in the laboratory identified by species.
Zunächst einmal kann man sagen, dass auf unserem Versuchsfeld die Regenwurm.
First of all, one can say that the density of earthworms in our trial field was very low from the start, so that we were not always able to take samples. For example, in 1 year we had to stop the sampling, because there were just no worms to be found. However, if one looks at the data we recorded, one can see that we were not able to detect any influence on the earthworms caused by the Bt variety. Do you know the reason for the low density of earthworms? Well, the low density of the earthworms from the start, before the experiment began and throughout the 3 years of the trial, certainly has to do with the field itself. The soil is rather sandy, and that's anything but ideal for earthworms. On the other hand, the agricultural use of the field, particularly the annual ploughing, will definitely affect the density of the earthworms. In the laboratory, Eva Schultheis performed a so-called earthworm avoidance test. The earthworm avoidance test is used to test for an avoidance reaction. It's not a test that we invented, but there is an ISO norm which proposes this test for ecotoxicological studies, and we applied it to our Bt maize and the cry proteins it contains. For the test, soil from the trial field was filled into pots. Half the soil was mixed with Bt proteins or ground up Bt maize leaves. The other half stayed untreated. In each case, 10 earthworms were placed along a dividing line. After 72 hours, the experimenter counted how many earthworms were in each half of the pots. We performed the earthworm avoidance test with a species that has been successfully used as a representative species in the laboratory in the past. For this species, we can say that no avoidance reaction was measurable. The earthworms were evenly distributed in both halves of the pots. This was the case for the untreated soil, for the soil mixed with one Bt protein or a combination of Bt proteins, as well as for the soil mixed with fresh or dried maize leaves. The concentrations that we used for the proteins were 100 to 1000 times higher than the protein concentrations in our trial field. So, in summary, we can say that an avoidance reaction did not occur.